Hello everybody, Namaskar. In this lecture today, we will discuss about probiotic and fermented vegetable products. Okay. We will discuss what are the probiotics and their health benefits, fermentation of vegetables, fermented vegetable products and also the bioactive compounds in fermented vegetable products. And finally, we will also discuss the microbial spoilage of fermented vegetables. Let us see what are probiotics. The probiotics are live microorganisms which, when administered in adequate amounts, confer a health benefits on the host. And most of the microbial strains which have probiotic activity belong primarily to the lactobacillus and bifidobacterium genera. They may be consumed either as a food product, like in the case fermented food product, or even this probiotic organism may be mixed with the non-fermented foods also, or as a dietary supplement, that is the products in powder, capsules, tablet forms. However, the various aspects concerning the safety, functionals and technological characteristics must be taken into consideration while selecting the probiotic microorganism. The development of a food product with probiotic effect has emerged as functional food intending to provide health benefits to the consumers, particularly to the mankind, and nowadays there is a trend toward making functional food or probiotic foods. Okay. So, it has to be seen that is because the food, when they are added with the probiotic microorganism, so when the food is processed, so the various aspect of the viability of this probiotic bacteria in the food products during the processing and even the post processing during storage, etc., that has to be considered properly. In fact, various factors which influence the probiotic strains in the food products are the type of the strain, type of the bacteria, the method of preparation of the bacterial culture, state of the cell inoculum, oxygen level, storage temperature, processing conditions, factors during processing like temperature and other things, and then the substrate condition of the food. In the food, what are the various nutrients, because it is important that the probiotic bacteria should be metabolically active at the time of consumption of the food, and for getting desired benefit, that a population more than 6 to 7 lakh CFU per ml or per gram of the product is required, that it must be ingested. Okay, for getting the physiological benefit. So, this is important, that is the probiotic bacteria in the food, this also, that is during storage and substrate, etc., should support the growth of, and it should remain in live condition. Okay. Then there are various reports on the health benefits of the probiotic food, etc. Okay. So, we are in this lecture, we are concentrating on non-dairy probiotic foods, etc. In fact, since the beginning, when this probiotic earlier it was studied that it was thought rather that only dairy product, milk and milk retail product will support to the growth of probiotic bacteria, because this lactobacillus, bifidobacteria, etc., that is milk is a good substrate. But now it has been seen, there are various researches people have conducted and which has proved that even non-dairy material like fruits, vegetables, etc., they also constitute a good substrate for the growth of probiotic bacteria. So, obviously, that is the various probiotic bacteria, they are selected properly, and in the I think non-dairy beverage or whatever substrate is there, fruit juice, vegetable juice or even concentrate, pulp, etc., in this it is added. So, when this bacteria is added here, then there are maybe that changes in the component, that is the fermented non-dairy beverage matrix okay, bioactives, etc. So this bacteria brings about some desirable changes into it. So when this food containing the probiotic bacteria is consumed, then once that the probiotic bacteria, it is live going into the system, and also the material which you are consuming, it also its components, characteristics are changed into this, that it has become beneficial. So in fact it gives a double effect inside our guts. These bacteria, they get established inside our gut, and where they modulate, regulate the functions. That is the secondary effect they provide, that this pathogenic bacteria growth, this anti-inflammatory, all those things are suppressed, and desirable microflora, that is grow, and the various desirable effects are produced inside our colon, inside our system, digestion and all those things. So it becomes in fact the various effects like anti-hyperglycemic, anti-fatigue, hypocholesterolemic, immunomodulatory, anti-diabetic, etc. All these various activities mean undesirable effects are suppressed and favorable effects are promoted. So accordingly, it becomes a very good healthy ingredient, that is consumption of probiotic bacteria or probiotic food. So now let us see common types of fermentation. In the earlier lectures we have discussed fermentation technology. So in this here, that the fermentation may be either alcoholic fermentation, which is generally encouraged by yeast, and it is mainly prominent in alcoholic beverages like wines, etc. In the last lecture we discussed detail about this. Then there is another type of fermentation, lactic acid fermentation, which is mostly done by, encouraged by lactic acid bacteria, and here major product is the lactic acid. And this is the major fermentation, you can say, in the fermented vegetables, fermented vegetable juices, etc. Then acetic acid fermentation is another, where acetic acid bacteria plays major role, which converts sugar into acetic acid in the sugarcane juice, etc. So the vinegar is the final end product. And alkaline fermentation also occurs, where Bacillus species are, and the products are known for their amino acid content, and this is mainly fermented soybean products. So when we are talking about fermented vegetable products, so basically this lactic acid fermentation becomes a dominating type of the fermentation process. So fermentation of vegetable, as I told you, lactic acid fermentation of vegetables and fruit is a common practice to maintain and improve the nutritional and sensory characteristics of these products. Fermentation, you have seen earlier, it is the oldest processing techniques which extend the shelf life of the food. Availability of certain specific nutrients like vitamins, minerals and acidic nature of the fruits and vegetable, it provides very conducive medium for fermentation of lactic acid bacteria. In fact, in some vegetables, etc., they contain also, that is, some oligosaccharides, etc., and which again becomes a very good food for the probiotic, and that is, they are considered prebiotic. So what I am trying to say is that these fruits and vegetables, they provide a good material, good substrate for the growth of both probiotic bacteria and other bacteria. Vegetables have low sugar content, but they are rich in minerals and vitamins and have neutral pH, and thus provide a natural medium for lactic acid fermentation process. So this obviously we have, in the earlier class we have discussed about the advantages of the fermentation. Here, to be very specific, that is advantages of the vegetable fermentation. Obviously it helps that in making the vegetable more nutritious, wholesome. Okay, it removes sometimes that is some of the anti-nutritional factors, etc., which might be there. It enhances the organoleptic and nutritional quality, retains the nutrients and colored pigments in the vegetables to some extent, and there is a prevention method for the production of finished and half finished product, and it prevents several diseases such as diarrhea, cirrhosis, liver, because of the probiotic properties, and fermentation based industries that are important source of the income and employment in the country, like several countries in Asia, Africa, Latin America, and so. Okay. and during the fermentation process the various metabolic and bioactive compounds are generated with some and which further increases the nutritional characteristics. So, here in this process a general process flow chart is given for making the fermented vegetable products. So, the fruits or vegetable after subjecting to the obvious treatment, that is the for cleaning, washing, cutting, grading, sizing, etc. Even sometimes whole fruit or whole vegetable can be peeled or unpeeled, they can be fermented. But so as the case may be, that fruit or vegetable that they are properly prepared. Okay. Sometime like in the case of olives, etc., that is they are size grading, and these olives are given alkaline treatment. Okay, then washing and neutralization, because they are to bring down that their acidity to in the label which facilitate the fermentation or growth of the bacteria. So, this is also done. So, depending accordingly that these mixes can be done, then blanching, cooking, tempering, etc. And finally, that is the prepared material. Okay, some if required in juicing or even in the cabbage, the shredding, etc. operations may be there. So, these prepared materials, they are added with sometime water or this sizes graded olives, etc., or other ingredient, that is sugars, starter culture, sodium chloride ingredients, other ingredient like spices, condiment, flavoring, etc. they are added, and all this together properly mixed or is smeared. Even sometime in some cases some nitrogen purging, etc. is also done to avoid the oxidation related problems. So, finally, it is subjected to the fermentation in suitable environment, suitable container, and then after the fermentation is over, the material is unpacked. It may be sometime even if required some brine can be added in infusion, or even this it can be filled or poured directly as a fresh consumed product, or it can be subjected to the treatment like blanching, canning, pasteurization, and pasteurized and fermented products packed in the proper glass containers or other proper now PET pouches, etc. are also used for its packaging. So, for marketability and convenience to shelf life. So, that is a overview of the overall general schematic of the fermentation, vegetable fermentation. So, let us see some technological factors of fermented vegetable products. So, obviously, to produce high quality fermented products, number one, the fruits and vegetable must be sound, undamaged, and at the proper stage of maturity. Raw material that are to be fermented, that is whole, must be suitably size graded to achieve homogeneous fermentation. The further pre-treatment depend upon the particular vegetable or its varieties, types, and it may include like peeling, blanching, cooking. Like in the case of green beans, blanching becomes important. In the carrots, beetroot, celery, etc., it peeling to facilitate a better fermentation process. Microbial action, the peeling might be necessary. Then cooking, alkaline treatment like green Spanish style, olives, etc., the alkaline treatment. So, suitable appropriate treatment is given. So, for fermentation, the pre-treated vegetables are placed in suitable fermentation vessel, okay, which may range from 100-liter drums to tanks or up to silos up to 100-ton capacity. Okay. In order to achieve favorable fermentation conditions, vegetables like cabbage, etc., should be shredded, and they must be salted very homogeneously on the conveyor belt between the shredding machine and the tank or during filling of the fermentation vessel. Whole fruits and cucumbers like olives and tomatoes must be placed into the vessels containing some brine in order to prevent mechanical damage and to displace the air between the vegetable parts. In addition to the salt, other additives optionally may as necessary may be added like spices, sugars, starter cultures, etc. The best practice will be achieved at fermentation temperature between 15 and 20 degrees Celsius. The fermentation time will depend primarily on the temperature, the type of vegetable, the degree of disintegration, and the desired acidity of the final product. Okay. The fermentation vessel or process, all this must be carefully monitored, and in particular it should be controlled for the development of total acidity and pH. to prevent microbial spoilage of fermenting vegetables, especially the growth of yeast and molds on the surface of the brine or within the fermenting products. That is should be taken care of. The addition of sorbic acid, tartaric acid, acetic acid and benzoic acid, these are permitted in several countries to take care of these unfermented vegetable or the microbial spoilage of fermenting vegetables. The final treatment product, that is the fermented product, its pH is less than 4.1. It must either be distributed fresh, packaged or unpackaged or pasteurized in pouches, cans or jars for later consumption. Ecological conditions in the fermented or fermentation vessel, that is the fermenting starter culture as well as substrate. The substrate is characterized by a solid in a liquid environment, brine, whereas the microorganism as well as the salt and their released nutrients can be distributed through the entire fermentation vessel. So, pathogens such as Salmonella, Clostridia, Listeria as well as other undesirable microorganisms, they are present in all probability. So, in general the raw materials used are rich in nutrients, growth factors and minerals. However, for microbial growth these substrates must be released from the plant tissue into the surrounding liquid, and the environment, ecological environment, this should be maintained as per the requirement, maybe water activity 0.95 to 99, pH 5.9 to 6.5 for cabbage and 8.5 is maximum for olives, temperature 5 to 20 degrees Celsius for vegetable, 25 to 30 degrees Celsius for vegetable juices, sugar content maybe 25 to 100 gram per kg of vegetable, buffering capacity 0.15 to 0.909 gram of lactic acid per 100 gram of vegetable, sodium chloride content maybe 0.6 to 2% for sauerkraut, 5 to 10% for brine of cucumber, etc. So, these are the indicative weights. So, as depending upon the material, depending upon the requirement, these conditions should be properly adjusted. So, the traits considered in starter culture for vegetable fermentation, there are three aspects. One is the pro-technological aspect, that is the starter culture. So, all the possible technological effort, that is it should be considered for selecting the starter culture, that is the velocity of the growth, how it grows, what are its requirement, okay, how it produces acid, what is its salt tolerance, okay, low pH value tolerance or completeness of fermentation process, okay, whether it is a mild acid producing or high acid producing, its tolerance to high concentration of polyphenolics, because most of these fruits and vegetables they contain more amount of polyphenolics. So, the starter culture should have good tolerance to the polyphenolics, then synthesis of antimicrobial compounds, pectinolytic activity, etc. So, all these things are the pro-technological consideration taken into consideration while selecting the that is the starter culture. And the second is the sensory characteristics, again whether it is a homofermentive metabolism or then it produces various products or synthetic of aroma precursor compounds, etc. That is what are the various secondary metabolites which are produced and how do they influence the sensory characteristics of the product, that is another very important consideration. And then the synthesis of nutritional and functional components, that is biogenic compounds, increase of the concentration of free amino acid, overproduction of vitamins, liberation of bioactive peptides, synthesis of the exopolysaccharide. So, all these are properly considered and the good micro starter culture is selected, and obviously if the starter culture raw material proper conditions maintained during fermentation. So, obviously it will give a novel fermented vegetable product. So, we will take one product, that is here one by one a few products, that is first we will start with the probiotic vegetable beverage with anti-diabetic potential. We have worked in our laboratory on this product. So, we have taken bitter gourd, carrot and bottle gourd. Bitter gourd, it has a bioactive compound present in it, is the polypeptide P momodicin, and it is known to lower the blood glucose level. So, bitter gourd is an anti-diabetic material. Then carrot, it is rich in bioactive compound like carotenoids and dietary fibers as well as natural antioxidants. It enhances the growth, encourages the growth of the bacteria. It provides a good substrate micronutrients for the bacteria growth. And then the bottle gourd, it is rich in vitamins and irons and minerals, so it is antidiuretic. So the three material we have taken in combination: antibacterial, antidiabetic, antidiuretic, and the one which is encourages the growth of the bacteria. And that, so one can take a single vegetable, but always it has been seen that if you take a combination of the vegetables, so you can have a multi effect health benefits, number one, and also the combination of vegetable may provide a better substrate, better suitable substrate for the growth of the bacteria. So we have seen that and this growth kinetics was studied in the MRS broth as well as in our vegetable juice, mixture of these three vegetable juice in appropriate quantity, and we have seen that, as you can see here, mu value, growth constant, etc., and almost mu, lambda, these are all comparable. So optimum parameters value for the logistic model describing the growth of Lactobacillus plantarum in MRS broth and vegetable juice at 37 degrees Celsius for 24 hours. These are given in this table, and they are all per table. So these data shows, and we have got a lot of data, everything in that produce here, that these vegetable juices, they provide good substrate for the growth of the probiotic bacteria. And the juice finally, which was fermented juice containing live probiotic, it no alcohol. Okay. So it has a good sensory quality, and its shelf life was found to be 60 days at 5 degrees Celsius temperature. So this in this is a process technology in brief, that is the bitter gourd, carrot juice and bottle gourd juice, that is bitter gourd 29%, carrot 41%, bottle gourd juice 30%, and its a TS content, total solids of bitter gourd was about 4.1% weight by weight, carrot juice about 6.4%, and bottle gourd juice about 3.68%. So these three juices having these recommended TSS, they are mixed in these proportions, okay, and finally inoculated with the Lactobacillus plantarum and our NCDC 414. That inoculation level was 1%, and the initial level inoculated was, that is the same log CFU per ml. So the fermentation on conditions are, the pH is adjusted, stressed that it should be 6, temperature is 31 degrees Celsius, fermentation time takes 14 hours. So sometimes it is there is addition of stabilizer, maybe 0.25% weight by weight volume, guar gum, then stirring at 300 RPM for 15 minutes, then salt is added and cumin seed powder. So this just to provide relishing effect and giving the good, improving the flavor, etc. further. So these are added, and then finally it is stored into transparent glass, you can see here, okay, and bottle at 4 degrees Celsius, you get a probiotic vegetable beverage, and that tastes very good, tasty and healthy beverage. Then probiotic carrot juice, that is the single. In the earlier we saw that carrot, bitter gourd or bottle gourd together. Here that is the carrot juice also is separately. This I had taken from the literature, that the process is same, that is the ground carrot, it is heated for 5 minutes at 85 to 90 degrees Celsius, cooling, and then inoculation with Lactobacillus plantarum, incubation 16 to 17 hours at 35 degrees Celsius. Then fermented carrot mass is obtained, which contains pH is about 4.5. Then there is a addition of enzyme, that in incubation for 60 minutes at 35 degrees Celsius, okay, and also citric acid is added here, okay. Then this is pressed for the getting the fermented juice, that is the pressing, filtration, and the juice is bottled and finally pasteurized 15 minutes at 85 degrees Celsius, cooled, and you get the pasteurized carrot juice. It is a probiotic carrot juice. Okay. Then sauerkraut, that is fermented cabbage, that is mature cabbage taken, wilting for 2 to 3 days, that is of course it is optional step. The removal of the outer and damaged leaves, washing, shredding, and then salt is added to 2.5% basis, and it is ensured that the salt should be mixed uniformly and properly to avoid spoilage, and then it is filled in vats, covered, the vat is covered with suitable cover, and it is placed by placing weight, etc. Weights of water are oozed out, that is so that the water oozes out from this, and then fermentation takes about 2 months at 20-25 degrees Celsius, you get sauerkraut. The culture here Leuconostoc, Lactobacillus, Pediococcus species are used. Similarly, fermented beetroot wine. Okay. Again, this is the same in the wine manufacture we have discussed in the earlier. So, almost same here, that is the similar taken, that is the beetroot juice is taken and it is subjected to the fermentation process, like the same is the fermentation, clarification, aging, etc. Okay. Then fermented pickles, that is these cucumber sized and cleaned flour removed, that cucumber can be taken, or other fruits or vegetable, whatever they are taken, washed, then brine is added, and this after addition of brine, lactobacillus strain, or even this acid is approved, that is with acetic acid, or it is sometimes purged with nitrogen to avoid oxidation related. So, fermentation continued, you can get salt stock, desalt, add acetic acid and package, or even genuine as such, package and refrigerate or even pasteurize. Okay. Then kimchi production process, in the earlier class also we discussed the details of the kimchi production. Okay. That is here it is the cabbage is using this process, it is the salted cabbage that is the brine for about 10-15% give us chloride for 10 hours at room temperature, and then sub ingredients which are there, red pepper powder, ginger, garlic, onions, etc. chopped, and then these are all prepared. So, in the spice lecture we discussed these things. So, a spice extract is obtained, this cabbage, and then these are two are mixed and fermentation is allowed, and the Leuconostoc, Lactobacillus, Vacillal, Octococcus, etc. are the commonly fermenting bacteria. Then other traditional fermented vegetables may be pao kai, Chinese sauerkraut is quite popular in Taiwan, like kimchi. The major ingredient is cabbage here, and the minor ingredients are garlic, onion, radish, carrot, spices. They are pre-treated and blended. They are then soaked in 6 to 8% brine for fermentation at 20 to 25 degrees Celsius for 6 to 10 days. Then torsi is a salted fermented product prepared from vegetables such as cauliflower, cucumber, celery, cabbage, Jerusalem, artichoke, and leafy vegetable like parsley, basil, tarragon. It is spiced with mint, garlic, hot pepper, and onion. The fermented vegetables are preserved in either vinegar or in the lime juice. Then bioactive components in the fermented vegetables, that is the lot of there are during the fermentation process, several organic acid are produced, that is lactic acid bacteria initiate a rapid and adequate acidification of raw material through the production of various organic juice. In fact, it is shown that the lactic acid and acetic acids are the commonly produced acid, that it also leads to the increased shelf life, these acid content. Okay. Even phenyl lactic acid is recognized as the major factor which is responsible for the antifungal activity that prolongs the shelf life of fermented vegetable, fermented foods. Then bacteriocins, another very important, that is the during when these bacteria grow, various lactic acid bacteria, etc., they produce various biologically active compound, biologically active proteins or protein complexes like protein aggregates, lipocarbohydrate proteins, glycoproteins, etc. They are produced. Okay. In fact, lactococcus, lactobacillus, pediococcus, leuconostoc, campylobacterium, streptococcus, enterococcus, bifidobacterium, all these probiotic and general bacteria, lactic acid bacteria, they are known to produce bacteriocins, and in fact, niacin is one such bacteriocin which is commercially produced by the bacteria, and it is used in several food industry. It has a very antibacterial activity, antimicrobial activity, has preservative, natural antibacterial. Then vitamins, several species of bacteria, yeast and fungi and algae, they may serve to produce folic acid, vitamin B12, vitamin K2, minoquinone, riboflavin, thiamine and other essential vitamins they are produced. So, these fermented products are good sources of vitamins. Even bioactive peptides, that is short chain peptide generally consisting of about 20 amino acids or so, these are also produced, they have the health benefits. Then exopolysaccharides, the most exopolysaccharide producing lactic acid bacteria belong to the genera Streptococcus, Lactococcus, Leuconostoc, Pediococcus and Lactobacillus. So, these exopolysaccharide EPS are also present in their fermented products and which acts as a prebiotic food for the probiotic bacteria. Then enzymes, lactic acid bacteria synthesizes diverse types of enzymes that impart the compositional, processing and organoleptic properties along with the overall quality of the fermented products. So, in nutshell we say these fermented products are a very good source. However, that is the care has to be taken. There are certain reports that microbial spoilage of the fermented vegetables may be there, particularly during cucumber fermentation. The formation and damage associated with the bloaters is a serious problem. The problem increases with larger cucumbers, higher temperature for fermentation and higher carbon dioxide level in the brine. It is therefore important to keep the carbon dioxide content in the fermentation mixture as low as possible. This can be done by careful control of any bacteria and yeast that produces carbon dioxide and removing the carbon dioxide from the brine using nitrogen gas. That is why in some cases that purging of the nitrogen is done. Circulation of the brine are limiting the growth of heterolactic fermentive bacteria which produces more and more carbon dioxide. Then this table gives that is a cucumber, that is there may be problem during storage, white or gray, internal spots, surface discoloration, softening, Bioactive formation, off flavor, butyric acid fermentation. In case of sauerkraut, discoloration, softening of or off flavor. Then cauliflower, discoloration, and in carrots, the bleaching, oxidized taste, etc. So, proper care should be taken to ensure that there is no microbial spoilage during storage. Then symbiotic food, that is the culture, and this symbiotic means now there is a trend toward developing the product like the vegetables. When you are adding probiotic, and this there are certain probiotic which produce the exopolysaccharides, etc. You have seen in the or sometime from outside some these prebiotic, that is the food of the bacteria, oligosaccharides, inulin, galacto-oligosaccharide, fructo-oligosaccharide, lactitol, lactulose, all these things. So, the probiotic bacteria and this prebiotic, that is bacteria food, they are mixed together. Of course, while selecting it prebiotic, the effort has to be taken that it should not adversely affect the sensory characteristics. So, these together. So, they are together, they were given when you ingest this symbiotic food, means you are taking the live bacteria as well as the food for the bacteria, and then it inside our gut this microorganism grow and give the provide a effect. So, one symbiotic beverage which again we have worked in our laboratory also, known as kombucha, that is also known as kombucha tea. It is a non-alcoholic fermented beverage brewed from sweetened tea and with a symbiotic culture of acetic acid bacteria and yeast, that is known as SCOBY, and also known as tea fungus. So, here that is the sucrose infused green tea or black tea is taken. Okay. Then this sucrose and this yeast enzyme divert, it acts and this converts it into that is acetic acid bacteria. It has some action on the cellulose. So, cellulose, gluconic acid, acetic acid, ethanol, carbon dioxide, all these are present, will be present in the beverage, and they give various effect. Okay. So, we have developed this technology like the water containing sucrose and tea, that is either black tea or green tea, which contains around 5 to 10% sucrose, and it here SCOBY is added, that is it is a previously fermented tea, the tea fungus SCOBY, it is 10 to 10% fermented tea, and these two are fermented. The temperature is around 20 to 30 degrees Celsius, up to 7 days fermentation is allowed to continue, and then you get that kombucha or tea. Okay. They remove the SCOBY and the fermented tea filter the liquid. So, bottle it can be bottle kombucha or junti, it is stored chilled or pasteurized. Pasteurization may be optional, or also it can be given ultrasonic treatment, maybe 750 watt power, 27 kilohertz frequency for about 20 to 40 minutes, and gives a stabilized kombucha or junti, and it can be stored up to 15 days in ambient condition, and it is a very good, healthy and tasty tea drink, that is fermented tea drink. Okay. So, with this I will summarize this lecture, that is novel preservation technologies. They have been used in combination with fermentation for certain fermented products, and there is still lot of scope for research and development in this area. Fermented fruits and vegetables, they contain a wide group of prebiotic components which attract and stimulate the growth of probiotic, etc. But a proper understanding for the microbial relationship between the microorganism, food, its constituents, beneficial effect, and the processing conditions during processing, how they behave, post processing, how the conditions are, how it is influencing sensory characteristics, its storability, etc. So, all these things are to be properly understood. Okay. And however, while preparing these fermented products, etc., the ingredients, when you are adding certain ingredient in the fermented foods like sugar, salt, monosodium glutamate, etc., it should be ensured that their levels in the end product does not increase the regulatory limit of the country. Okay. Because sometimes the sugar, salt, nowadays there is a trend for consuming low sugar food, low salt food, and all those things. So, so with this I thank you very much for your patience here. Thank you.